Good afternoon, everyone. My name is Sharon Rounds, and I am the PI for Advance-CTR, which is an NIGMS funded partnership to enhance research infrastructure here in the state of Rhode Island. This year, we're very pleased to be offering our translational research seminar series, which is focusing on really outstanding investigators in clinical and translational research in the state. And today, we're very, very pleased that Jill Maron is with us. Jill is the Pediatrician-in-Chief at Women and Infants Hospital, and also a professor of pediatrics at the Maron Alpert Medical School. She holds the O. Zopfi Endowed Chair for Perinatal Research. Jill came to us from Tufts about a year ago, I think, and where she was vice chair for pediatrics research and executive director of a mother-infant research institute there. She did her undergraduate education at Harvard and received the MD and the MPH from Tulane University. She was a resident at Hasbro in pediatrics and then went to Tufts for fellowship training in neonatology. So it's really been a wonderful pleasure to welcome Jill back to Rhode Island. So her research has focused on the identification of biomarkers in non-invasively obtained biofluids, and this will be interesting to hear about, and the subsequent development of diagnostic assays for use in these delicate neonates. She serves as PI on an NIH-funded multi-center award that integrates rapid genomic testing platforms into the care of critically ill newborns. Her research has been funded by the NIH for many years. She's published extensively. She chairs an NIH study section, and she's a co-editor of translational research, I believe. And so it's just a real pleasure to have you here today. And Jill will be talking to us about her work in developing and integrating novel diagnostic platforms into newborn and perinatal care. And as noted, please enter your questions into the chat and we will address them at the end of the presentation. So thank you again, Jill, for joining us today.
Well, thank you, Sharon. It's really a pleasure to be here. Thank you for the kind introduction and the opportunity to share my work with the group. I have no disclosures to make. But today, what I want to do is share my journey, which has really been over the past 15 years, that is centered upon the development and integration of neonatal and perinatal diagnostic assays. My goal today is to share how I've approached development of these assays and new areas of discovery. And importantly, I hope to excite you, my colleagues, about the potential to integrate diagnostic platforms into clinical care to improve our outcomes. It's an exciting time for all of us right now when we look around at the emerging technologies that we can utilize to care for all of our patients, whether we're talking about miniaturization of data chips, all the biosensors that are coming out. Some of you are probably wearing them right now on your wrists. The scale of which we can actually analyze network biology and understand the complex interaction of biology, combined with the speed with which we can get that answer, which is largely being driven by artificial intelligence. And I would argue that technology right now is not our limiting factor for improving health outcomes. The technology is emerging far more rapidly than our own understanding of the biology of the patients and how to apply this technology in a meaningful way so that we can actually impact care. This is my patient. This is what I spend my time in the NICU. These are my very fragile patients that I take care of. And it's just looking at the picture, you can imagine that I have many challenges associated with them. But for those of you who aren't familiar with neonatology, in 2021, this just came out this past month, the March of Dimes Report Card 2022, 10.5% of all pregnancies in the United States right now result in preterm birth, which is defined as anything less than 37 weeks gestation. And there's approximately 15 million babies born prematurely worldwide. And while the majority of these babies are considered late premature, which means they're born between that 32 and 37 week window, the current edge of viability right now it hovers at about 22 weeks gestation. And in fact, at Women and Infants, we have babies at 22 weeks gestation that we care for. These babies have many challenges, and I'm just going to orient you to their blood volume. On average, we all have about 80 to 100 cc's per kilogram of blood, but my babies often don't even come close to weighing a full kilogram. So a 500-gram infant, which is really pictured here, has only about 3 tablespoons of blood in their entire body. So obviously blood is a very precious commodity, and we want to limit the blood that we take from these babies, which is often a challenge because we have to do frequent blood draws. This is their skin. It's extraordinarily fragile and is translucent. It can burn easily. So those emerging biosensors that are coming out are really still problematic in these populations. The goal would be, wouldn't it be wonderful if we could put a biosensor on a baby and do things like continuous glucose monitoring or analyte monitoring? And while I think the field is moving towards that, it's still not that easy for these babies just because of the fragility with which they have. And of course, every time we get a sample, it's a challenge. We often use heel sticks to get blood if we're checking blood glucose, for example, but this is how we get CBCs from these babies as well. And this doesn't come without a consequence. It's well known in the field. This was published in 2016 by Gokular that comparative heel stick studies showed that newborn infants who had undergone repeated painful procedures showed increased short-term pain responses. And we see this over as they grow up. They will continually complain about certain pain and their pain sensitivity. And of course, enrolling these babies into clinical trials is a challenge. It goes without saying they're highly vulnerable when you're trying to integrate a new diagnostic, assay, drug, test. There could be unknown risks or benefits with the treatment. You're approaching families for enrollment at an extraordinarily vulnerable time in their lives. And of course, the parents have to consent. They have to be willing for their babies to participate in these trials. And what's happened over the years is that we've had a paucity of anything coming out that's been approved for drugs. If we look at just pediatric progress from 2003 to 2017 at the FDA level, you can see in this inverse pyramid just how few drugs and drug change labels for neonates have been forthcoming, even in such a long period of time of almost 15 years. And then these babies are rapidly developing, and I don't want to underestimate the impact that has on biological variability in this patient population. If I were going to introduce a diagnostic assay into adults, I could think about it in years, but I have to really think about this in days to weeks for a baby, especially one that's born premature. Here's just a schematic of how rapidly their brains are developing, and all their organ systems are developing that rapidly, including the kidney. We know a baby who's born at 22, 23 weeks gestation, that kidney can't hold on to big analytes. It just dumps it all into the urine. But over time, it gets much smarter. Same thing with the salivary gland, which is where I focus most of my work on. What's filtering through the salivary gland from the blood into the mouth changes over time as that gland develops. And we have to remember that a 24-week infant is not the same as a 26-week infant or a 30-week infant or a 36. And this is a huge challenge in terms of developing these assays. But I had to think about this in a way that I could sort of overcome multiple challenges that we just discussed. I needed to think of something that was safe, non-invasive ideally, Caregivers would buy in, and more importantly, patients, and by proxy here, the parents would buy in. I needed to be able to leverage technology that was emerging to analyze those analytes, even though it was going to be very limited blood or salivary volumes, and I'd had difficulty accessing it. And I had to be able to begin to account for biological variation so that I could discriminate between health and disease. And I thought perhaps the answer just might be saliva. So saliva in neonates, really utilizing this biofluid, this is probably the perfect patient population to do it. They always have it. They're going to make more, and I never hurt them by acquiring it. I avoid phlebotomy completely, so I'm not taking blood from them. It doesn't hurt them, so there is none of that noxious stimuli that we get from blood sticks or needle sticks. And parents, I have to say, almost always say yes. If I approach a family and say, I would like to collect saliva, is that okay? Most families will say it's not a problem at all, have at it. And if you don't know, this is a rapidly emerging field, but it actually dates back centuries. It was the Chinese cultures hundreds and hundreds of years ago that said saliva is the mirror of the body, and whatever you can actually find in blood is truthfully sitting in the mouth as well. When we look across where the field is currently, commercially, it would take you all of 2 seconds to find multiple diagnostic companies that are developing collection kits for saliva. These can be used both in the home, they can be used in a research setting. Everything from parents could drug test their teenagers at home. There are HIV tests at pharmacies that use saliva, and of course, the genetic boom of 23andMe and Ancestry.com has taught our country and the world that you can spit in saliva and learn an enormous amount about yourself, your heritage, and your relatives. And when we look on PubMed, all of these can be found for salivary diagnostic assay development, but what you'll see is that none were targeting the newborn. So how we could integrate emerging technologies into neonatal care really became dependent for me upon 4 fundamental aspects for success. I had to answer 4 questions, and the first was feasibility. This concept of, can I even do it? When I talk about small volumes, I mean small volumes, 1 drop, you can barely see it in the collection device. If I can do it, what's the need for it? And who are the stakeholders that I need to have to buy in so that I can translate it? Is it reproducible? Does it work? Does it work across many NICUs, diverse patient populations? And then is it reliable? Is it actually accurate? Can I trust the readout? What you noticed probably was missing was I didn't mention a biological mechanism. That is in some ways deliberate, but I don't want to give the impression that I don't think biological mechanisms are important. They actually are the foundation of the work I'm about to present to you. But in the neonatal population, assessing biological mechanisms is very challenging. How are these biomarkers getting into saliva? What are their mechanisms of action? I'm not going to be able to answer those questions for you. Rather, I use preclinical animal data, unbiased network analyses to target biomarkers that I'm seeing repetitively in saliva that are correlating to the phenotypes, the health conditions, or the medical concerns I'm looking at. Additional neonatal considerations, again, the small volumes, can it actually provide relevant information? And if anyone's ever done a salivary test, you'll know that you're told don't eat for 30 minutes. Swish and swallow, and to clear the oropharynx. And what you're trying to do is get rid of food, debris, any other contamination. Impossible to do with a newborn. You can't do that. They all reflux, every single one of them. So you're always at risk for having something like breast milk contamination, and that's real. And so we have to think about that when we're interpreting data and making sure that reliability and reproducibility will hold. How I actually collect saliva is pretty easy. Although there are a wide variety of collection devices on the market, when I first started, no one thought about a newborn. So I had to sort of come up with my own plan. And it's pretty simple. What we do is we take a 1 milliliter syringe that we get. We actually pull out the plunger here and we cut with scissors those end caps. And the reason we cut it, because I want a cylinder so that I can just attach it to low wall suction. And then I gently suction the mouth. And when I tell you we only fill that hub, I mean, we only fill that little hub. You'll get 1 drop of saliva in there. I then put the plunger back, I stabilize it at the bedside, and we bring it to lab. There are sponges out there that you can use for neonates. They do have some benefits. You have to understand what you're looking for in saliva. The sponges are going to remove cells, microbes, mucins. All of this may be helpful, but you just want to have a good understanding of what you're collecting and what's the potential limitations of it. When we started this work, we would take that saliva and we would say, well, what do we have? What do we have in terms of quantity for volume? We would do this by weight, and we knew we were getting about 10 up to most 50 microliters per collection. We would quantify what was in there. My work really focuses at the RNA and protein level, and we would get around 500 nanograms of total RNA and protein. And then we would look at the different levels of that saliva. Even though it was a drop, it was stabilized in a fluid solution, so we could spin it down to get those cells down into a pellet and look at the supernatant. We knew, however, that that yield was really small. And then we use something called a bioanalyzer, which can do both a quantitative and a qualitative assessment of RNA specifically. And we could see, what are we actually getting? I want to highlight here that I'm not interested in cellular RNA from the mouth. I'm not interested in what the tongue or the palate or the cheeks are transcribing. This concept that blood enters the salivary gland, is filtered, and then sends signals into saliva, that's what I'm targeting. That's that whole body picture that we're going after. What we know is that that is absolutely degraded in all of our mouths. If you look at the supernatant, it's going to be more degraded than whole saliva because it doesn't have cells. If you look at cell-free RNA, it's going to be more degraded than cellular RNA. And this is what it looks like. These are actual neonatal salivary samples. This is your supernatant here. And this is your whole saliva here. And what we're looking at total RNA, if you remember, it's mostly comprised of ribosomal RNA. You see 18S and 28S of humans. Here's your total RNA. These are your peaks. This is whole saliva. It looks quite nice. The area under the curve is the amount of total RNA present. Supernatant doesn't have that. Okay, it is very limited. These are your peaks that you're seeing. So we knew right away that supernatant is possible. There's a tiny amount of RNA that can be analyzed. Whole saliva is going to be better, but it does contain cells. But it was feasible. We could do it. Just that 1 drop was going to give us something. So we had to go figure out what we were going to get. So despite the limited quantity and quality of salivary samples, neonatal saliva could be successfully collected, processed, and ultimately analyzed. So what was the need? Saliva provides us, and I hope I convince you of this by the end of the talk, an unprecedented opportunity to assess real-time events in this population, both in terms of development and health status. We knew key stakeholders, as I told you, were going to say yes. Both parents and caregivers were really interested in what I was doing at the bedside. But that biological variability is real. And so as I went to develop these assays, I couldn't go after rare morbidities. It wasn't going to be helpful. I never was going to get there. I would have to screen thousands and thousands and thousands of babies just to get those rare morbidities. And it really wasn't something that I was going to be able to make an immediate impact on in the field. So I went after 2 specific areas of investigation: how babies learn to eat, and are they infected? How babies learn to eat, I targeted because every single baby in that unit is trying to do it. I wanted to know if you could do it. I wanted to know if you couldn't. And when I walked in that NICU, every baby was up for grabs for me. And the same held true for infection. If you're in the NICU, you're most likely born premature, or you're already sick because you're a term baby. You most likely have either IVs or central catheters, and you're all at risk for infection. So again, it was almost every baby when I walked into any unit that was eligible to give me saliva to understand these 2 conditions. So as we talk, and as I was thinking about, well, how do I develop a panel? There really are two approaches. The first is I could start from square one and say, I actually don't know what I'm looking for in saliva. That's my biomarker discovery approach. And I'll take a condition, and I'm going to use large scale screening, such as sequencing, to identify potential targets in saliva that could discern conditions. For example, I can feed or I can't feed. The other approach was I already know the biomarkers I want. I just want to know if they're in saliva, if they're quantifiable, and if they're informative. And that's a much more targeted approach. So the two panels that I've worked on, one involves biomarker discovery, and it is related to feeding. As a pediatrician, and we can't help ourselves, we all have acronyms. I developed the NOURISH panel. NOURISH stands for Neonatal Oral Feeding Readiness and Salivary High Throughput Diagnostics. And this was an R01 funded multi-center trial that just came to completion. The other one was utilizing known biomarkers, and that's the SPITS panel, and that's salivary profiling in infants treated for suspected sepsis. And this is an ongoing R01 that's currently being conducted across the country. So let's start with neonatal oral feeding. Why did I target this beyond the fact that every baby has to learn to feed? And it really was simple. You know, if you're born premature, you don't know how to eat. You actually have to learn how to do so in the NICU before you can go home. And our ability to assess an infant's readiness to try feeding is somewhat limited. It's based on bedside assessment, and that's pretty much it. Is the baby alert, awake? At a developmental age, we think the baby may be able to feed, not working hard to breathe. What we really struggle with is, all right, well, I tried to feed the baby didn't eat, but I don't have a clue why. And then I wanted to use that information to actually personalize treatment strategies for that individual baby based on what I was seeing in their development. Most of us think of oral feeding as simply, well, if I can suck, swallow, protect my airway, and breathe, I'm good to go. And it turns out it's far more complicated than that. And I spend a lot of time thinking about feeding, but I'm going to take a moment to share with you some of this. Our ability to eat isn't just, can we suck, swallow, breathe, or chew, swallow, breathe? It's actually highly driven by our senses. And we know this. We heard a lot about this during COVID. Everyone who lost their sense of smell couldn't taste, and that's true. And if you can't do that, your pleasure of feeding really isn't there anymore. And this plays a huge part in how babies learn to feed. They know where their mother is. They actually can discern their mother's breast milk from someone else's breast milk, and those studies have been done. You also have to know you're hungry. That gut-brain axis is serious business. We know when to eat. We know when to stop eating. Babies don't have a clue, particularly if they're born very early. They actually would sleep all day long. We feed them. We make sure they get fed, but they're not waking to feed. But before they go home, or a term baby is going to wake every 3 hours, scream his or her head off, and demand to be fed. And that's coming in right here. That's that gut-brain axis maturation. And then finally, neurodevelopment. That's all being coordinated at a neurodevelopmental standpoint. So when I started here, I said, all right, I'm going to get as much saliva as I can, and I'm just going to do something really simple. I'm going to compare what the saliva sample of unsuccessful oral feeder looks like compared to a baby that absolutely knows how to do that. And I used RT-qPCR and microarrays to look at the difference. And this is how we got NOURISH. We ended up, after looking at 360 babies, a comparative analysis, identifying a panel of 5 genes that were brought to the forefront that were involved in diverse developmental systems in feeding. 2 were involved in hunger signaling, 2 were involved in sensory integration, and 1 was involved in facial development, that importance of, can I structurally suck, swallow, and breathe? And what we learned in this work was the following. Of these genes, if you were a successful feeder and I looked at your saliva, I could detect these 2 genes, AMPK, which regulates whole body energy balance, and activation of the gene in the hypothalamus induces feeding and waking, and plexin A1, which controls axon guidance, and we see increased expression in the mature compared to developing olfactory sensory neurons. So if a baby could eat, I could detect both of these genes. At the same time, if a baby could eat, they had downregulated these 3 genes. One was MPY2R. This is also a hunger signaling gene. And it turns out we downregulate that to feel hunger. So that's a gut-brain axis gene. And when our body says it's time to eat, we're going to downregulate its expression. Wnt3 is an embryological gene that's involved in lip, palate, and tooth formation. And once that area of the face is formed, that gene should start to turn off. And NPHP4 is involved in retinal development and visual behavior. Again, as the eyes develop, that gene's going to turn off as well. And so here we were, our successful feeders had turned on Plexin A1 and AMPK, turned off these three. This is a sensitivity, specificity, positive, negative predictive value, odds ratios, and p-values of each gene. We were a little liberal with our p-values and incorporating that into the panel. I do want to highlight two things. First is that age matters. As a baby matures, the more likely they are to become a successful feeder. And what neonatologists have known clinically for a very long time, girls actually learn to feed sooner than boys. And we saw that clinically. This has been well published as well, and it held true in this particular study. But you'll notice we don't have anything that's really a standout. AMPK has pretty good sensitivity, and Wnt3 has pretty good specificity. But there was no magic bullet. And we knew that going in. It's because feeding's complicated. It wasn't going to be one developmental system that dictated whether a baby could eat or not. It was going to be the combination of them. So that's what we did. We combined all of the biomarkers together, and then we randomly selected samples from the data set to generate a receiver operating characteristic curve. And if we did that and asked the question, well, how good are we? If I got saliva and looked at all 5 of these genes, how good am I at predicting whether a baby can feed? And we were fair. We weren't great. We were fair. Sensitivity here, 1 minus specificity here. Reminder, this is no better than flipping a coin. And our area under this receiver operating characteristic curve was 78, or we were about 78% accurate. But that wasn't the power of the test. A nurse could have told me whether a baby could feed. She'd feed, come out and say, yeah, the baby ate, or no, the baby didn't. The power of the test was, why didn't the baby eat? So let's look at it a different way. I told you 3 genes had to be off for a baby to feed, and 2 had to be on. And if I took a 35-week male and I saw that profile, that baby had a 60% chance of eating. This is how a girl does it. Same thing. I've done nothing but change it to a female. She was 73% chance of eating. But what if I could understand why? What if I just said, okay, well, let's pick one gene, Plexin A1. What happened to those babies that had everything right, but their Plexin A1, their olfactory wasn't developed? What happened to them? Well, they dropped to 35% feeding. And here's where the power of the assay was. It wasn't just that you could tell me that baby can eat and that baby can't eat. It was the first time we had molecular evidence that said, I think that baby can't eat because we have a sensory issue. That olfactory system isn't there yet. And why that matters to me as a caregiver, because we do things like kangaroo care, where we put the baby on the mom or the dad, sometimes for hours, and that's a huge boost to their sensory integration. Or we ask the mom to leave something of hers at the hospital and we put it in the isolette or bassinet. These are strategies that we know can help feeding, but this told me what baby perhaps it's going to help the most on. But as good as this was, I knew I had more work to do. You saw the predictive value, 78%. So there was still a gap in knowledge regarding essential molecular mechanisms that I was missing. So I then went to RNA-seq and thought, let's use this platform. Could we improve our understanding of oral feeding? Again, this is all hypothesis discovery. I don't know what I'm going to find. I don't know what these genes are going to be, but I'm hoping that RNA-seq is going to allow me to develop novel pathways related to oral feeding success that we haven't seen before, so that I can further optimize personalized approaches to feeding outcomes. So we did that. We did RNA-seq on saliva samples collected. Just again, you can feed, you can't feed. And we made every attempt to match for the age that the babies were born, the age at when we collected saliva, their sex, because it mattered, and race and ethnicity. And we looked at those data in a comparative and systems biology analysis to look at differentially expressed genes, both between those who could and couldn't feed and males and females. And if we just looked at the group at the whole, we weren't bad at discrimination. There's some overlap, but what we see here, this is a PCA plot. The yellows are my successful feeders. My blues are my unsuccessful feeders. They cluster, but not perfectly, which is appreciated here on the heat map. Here they are, and there are some overlaps. These are the 63 genes that were differentially expressed between those babies. 59 mapped to known gene functions and 4 didn't. And so, of course, what we did then was we said, well, what do those genes do? Where are they? What's going on with those genes? And this was really important because when we went to RNA-seq, we were both able to validate what we had seen previously on those other platforms and identify novel networks that we had never seen before. And the novel network we had never seen before was memory. We had seen a lot of hits coming from the hypothalamus, but this was the first time we saw hippocampus. And of course, that makes sense. You can't feed if you can't remember how to do it. And so this was the first time that we started to see genes involved in this system. We were validated again with palate. That was a really important structural gene that we needed to see, and sensory integration again. And it was the same too. It was vision and smell that we saw. So we ended up modifying the Nourish platform. We threw in memory. The gene here was CDH13. We kept Win3, but added a new one known as FOXP2, which I had done some other studies on. We stuck with MPY2R and eliminated AMPK because it really is ubiquitously expressed, and we wanted to sort of more targeted go to the hypothalamus, and we kept our vision and our olfactory genes. And then I paired up with a neuroscientist. This is Dr. Stephen Barlow. He's the department chair of neuroscience at the University of Nebraska-Lincoln. And Dr. Barlow invented this machine. It's called the N-Trainer, and it's FDA approved. And you just bring this machine to the bedside, and you actually work in a very safe manner to teach babies how to suck. Again, babies can suck on a pacifier. It's just a rhythmic reflex that they can do. And it's very different from suck, swallow, breathe. And so this machine trains a baby to go from I can just suck on a pacifier, I don't know what I'm doing, to I can actually eat. And you can use this machine anytime you want at the bedside, and you can both assess how the suck is developing and actually do an intervention. So Stephen and I combined these. I used his machine at the bedside with babies, and I collected saliva to see what impact we were having developmentally when we did this. And this was a 5-year NIH-funded multi-center clinical trial, and it assessed both how the N-Trainer was working, along with what the Nourish platform could tell us in a cohort of infants that were born very premature, so less than 29 weeks. And we wanted to look to see, does the N-Trainer improve their oral feeding outcomes? And could we understand the developmental complexities of oral feeding at a molecular level when we did so? And I write that this is one of the first neonatal salivary diagnostic clinical trials on clinicaltrials.gov. It actually may be one of the first. It may be the first, I should say, but it got up there. And so how we did it was we randomized babies to receive either the intervention or just the assessment, which is called sham stimulation with the N-Trainer. We collected saliva samples throughout the treatment and the learning process of oral feeding to leverage this new Nourish platform. And it was our very first attempt to understand how genes were changing in response to therapy. Salivary collection occurred immediately prior to the initiation of gavage feeding. And that's gavage feeding, where we have a tube that goes in the baby's nose down to his belly because he doesn't know how to eat. And then we did the N-Trainer during an actual feed. Okay, and that's important because I think that had the most impact on our results. We didn't just randomly do an N-Trainer. We did the N-Trainer while the baby was simultaneously getting fed into his stomach. And what we found was that infants who received the N trainer learned to orally feed at an earlier postmenstrual age than infants who received the sham. And interestingly to us, and I have to say, I wasn't sure we would see this, but salivary profiles revealed that this intervention impacted sensory integration and hunger signaling, not oral motor maturation. So even though the machine was designed to do that, at a molecular level, we weren't impacting that system. We were impacting sensory and hunger signaling. And we speculate that it was because they were receiving the intervention during an enteral feed that was why we're making such an impact there, because the infants correlated feeding as well as hunger and sense of satiety with the oral training. There are numerous manuscripts in preparation for this trial. They will be forthcoming, so stay tuned for that. But I want to share with you what we were doing simultaneously. The strengths of this study was that we could monitor diverse and emerging developmental systems. It was the first study to assess feeding intervention at a molecular level, and all of it was done in a benign fashion. But let's go on to infection. Our ability in the NICU, particularly, to discriminate between babies who are infected and not infected is a major challenge. When you are in the nursery or the NICU, we are really the veterinarians of medicine. Our babies cannot talk to us. They all forget to breathe half the time. We don't know if they're infected or they're just having a bad day. And so we get scared because infection can very quickly kill these babies. So the majority of babies exposed to antibiotics in the neonatal period are only suspected of being infected. And in fact, they were never infected at all. So antibiotics are the most prescribed drugs in the NICU, despite the fact that most newborns are not infected. Goes without saying, we know that's probably not good, right? Exposure to unwarranted antibiotics increases microbial drug resistance. It may alter the microbiome. And in our babies, that microbiome is just forming. And we know from animal studies that once your microbiome is formed, it's very hard to change it afterwards. We realize this is a problem, and I will say across the field, we're doing our very best to have increased antibiotic stewardships. We've developed specific neonatal algorithms to say who should and shouldn't get antibiotics. And of course, the field as a whole, not specific to neonatology, but has developed novel multiplex PCR platforms to quickly identify an organism, to improve our diagnostic accuracy, and to reduce antibiotic exposure. The other concept you could use are inflammatory biomarkers or cytokines. Neonatologists either love or hate these, I'll be honest. Some think they're great, some think there are ways to even look at. So it is a hot topic in my field. But there's no doubt that if you are infected, you're going to mount an inflammatory response. And even my tiniest babies will do that. 24-week babies will skyrocket their cytokines. But in order to use cytokines in an informative manner, you really have to follow them serially. A single cytokine biomarker is probably not going to be specific enough. And right now, you rely on blood sampling to do that, and a lot of blood sampling to do that, because many of those cytokines are send-downs. So that's really not feasible for our babies. But we had an idea. Could we develop a non-invasive, ultra-sensitive salivary diagnostic assay to improve infection screening that wasn't based on microbial detection, but was actually based on host response to infection based on cytokine levels? And we got that idea because way back in 2014, my group was the very first to recognize that salivary CRP was detectable in neonates and that it actually correlated to blood levels. And we published this way back in 2014. You can see again here an area under the receiver operating characteristic curves. If your serum CRP was greater than 5, this was your correlation in saliva, and we had improved correlation if your serum was even higher, greater than 10. But this is limited. We couldn't just use serum, the salivary CRP. It was a singular cytokine. We needed to develop an assay that could look at multiple cytokines. And I really wanted it to not hone in on a single infection. Sure, a baby may have a bloodstream infection, but they could have a UTI, they could have pneumonia, they could have a viral infection, they could have meningitis, they could have a cellulitis. I wanted something that said, you do have an infection somewhere. and you should be on some type of therapy. And I wanted to be able to monitor those cytokines over a short amount of time. So here I partnered with Dr. David Walt at the Weiss Institute at Harvard Medical School, and Dr. Walt had invented the single molecule array, or what's known as Simoa. It's commercially available today, but it has limits of detection in the sub-picogram to milliliter range. It's greater than 1,000 times more sensitive than ELISA's. It can detect up to 12 cytokines from a single saliva sample. All analytics are performed on a single chip, and from salivary acquisition to a readout, it takes about 45 minutes. So this was SPITZ. This was our trial. Right now, it's a multi-center R01 trial. It tests the hypothesis that serial salivary profiles within the first 36 hours of infection workup will correctly discriminate between infected and uninfected newborns. We're not, again, targeting a single organism or type of infection. We are quantifying 12 cytokines in a single drop of saliva, and Women and Infants Tufts and the University of Florida, Gainesville are participating sites. I have to do one thing. I'm going to apologize. This particular slide does not want to show, so I'm going to apologize for that. So I want to show you what we're seeing. This is preliminary, okay? This is preliminary data. We've over 1,000 infants enrolled currently. This was the first 330. I want to show you that here we are. This is what we're looking at is IL-1 beta 6/8, TNF alpha CRP protein, 12p70, IL-4/5, interferon gamma 22 and 10. What we're looking at here are septic babies, clinically infected. These are babies that the physician has said, I know my blood cultures are negative, but I don't trust this baby and I'm going to give him antibiotics. And then non-infected. Each dot is a level, okay? And what I want to highlight here is one, we can detect all 12 in a single drop. Here's your biological variability. I want to say I haven't normalized this for anything, not age, not sex, but here it is. You see it. And you also see that most babies aren't infected and all of them got exposed to antibiotics. But we do see mean differences across many of these cytokines. And this again is all done in one drop. And when we model this out, when we're training the model, our accuracy is about 87%. When we take it to full, it does drop slightly below, but we're above that 80% threshold of predicting who is and who isn't infected. And we can actually say, well, which cytokines are the most important? What we're seeing in both models, IL-4 followed by CRP, interferon gamma, and so on down the list. So this work is absolutely ongoing. And the only reason I wanted to show you that is just to show you what we are able to do in today's day and age to detect these cytokines, and that hopefully they will actually be highly informative for us. All right, so what about other areas have I worked on? I'm going to focus on two. Neonatal abstinence syndrome, which is when babies are born exposed to opioids in utero and undergo withdrawal. I've also looked at speech emergence, and that is really because how we feed is also how we speak. And so we follow these babies out to 2 years and monitor their salivary expression profiles. And then I'll end with placental biomarkers in maternal saliva, particularly as it relates to preeclampsia. So if you don't know, we have an epidemic in this country, particularly opioid use globally, but even in pregnancy. And sadly, every 15 minutes in this country, a baby is born who has been exposed to opioids in utero. Babies who have been exposed to opioids in utero and have what's known as neonatal abstinence syndrome feed in a very unique manner. They first start out uncoordinated, and then they don't stop eating. They have hyperphagia. We typically think of a term infant can consume 80 to 120 kcals per kilo per day, but infants with NAS will consume 150 to 220 kcals per kilo per day, or even more. And the dogma always was that excessive caloric intake is because they have increased caloric demand, because they are withdrawing. They're shaking, they're breathing fast, they're irritable, they're crying, and they're burning more calories. But as you know, I study feeding, and those biomarkers didn't just appear. I had to study this for a long time. This is your hypothalamus. This regulates feeding. Makes you feel satiety. These pathways make you feel hunger. But I want you to hone in here because this is your arcuate nucleus. And you've heard me mention some of these genes. Here's MPY and here's MPY2R. Okay, so this is regulating hunger. We'll see the leptin and the leptin receptin gene. You may have heard of ghrelin. All of these regulate hunger. But let's go over here because this is your cocaine and amphetamine receptor. This is your opioid receptor. This is how SSRIs work right here. Okay, this is where naltrexone works as an antagonist to your mu opioids. They're all sitting together. So all our hunger signaling genes sit right next to all our drug reward seeking genes. And I thought, what's the connection? Could there actually have been a developmental disruption in reward-seeking behavior in NAS babies? And was it possible that these babies were overeating because food was eliciting a similar response to opioids? In other words, as we were weaning off opioids, was food becoming their drug? And could there actually be a sex-specific response to in utero opioids? We already saw that girls feed earlier than boys. Well, it turns out boys are more likely to withdraw from opioids compared to females. So we did a pilot study with this wonderful mentee of mine, Dr. Elizabeth Yen. We did a prospective case control observational study where we collected saliva samples from subjects within 48 hours of birth prior to them ever needing treatment for withdrawal. And we actually just did simple RT-qPCR assay looking for our friend MP2YR, that hunger signaling gene, along with two others, POMC and the leptin receptor. And then the most well-studied reward signaling gene, which happens to be our dopamine DR2 receptor. We normalized appropriately, and we used something called the delta CT method to quantify the analytes. We recruited 103 subjects. There were 3 sample failures. So we ended up with 100 subjects enrolled. There were 50 controls and 50 cases, and they were matched well. So 27 males and 23 females in each. The males, no surprise, required treatment more than the females. No pharmacy for 16 and no treatment for 18. And what we saw when we said, okay, well, let's group them all together, cases and controls. Here are our genes. Here's our delta CT level. I just want to say it's a PCR thing, but delta CT levels are inversely related to overall gene expression. So in other words, the lower the level, the more you're expressing, okay? But there was no difference. There was nothing there. So then we said, okay, well, what if we look at treatment versus no treatment? So you were exposed to it. What do you look like if you were treated or not treated? There was nothing there. So then we said, okay, well, let's split it up by sex. No difference in the controls, but there it was in the cases. If you were a male infant, you were much more likely to have higher expression of that reward-seeking gene than the female counterparts. And then we could also look at that based on whether you needed treatment or not, and it held true. The males were much more likely, if they didn't need treatment or if they did need treatment, to express the reward-seeking gene than their female counterparts. In the treatment group, males also express the leptin receptor more. And then we could actually go on and correlate that to volume intake, okay? So when we looked at how much volume these babies were taking in each day, again, knowing that this is a higher amount of expression, we could see both on day 2 and day 7 that DRD2 here in blue was correlating to the amount of food you were feeding. So these data really suggested that hyperphagia seen in infants with NAS may be due to developmental disrupted reward signaling pathways where food becomes the drug, and that there are sex-specific differences, and we could finally see perhaps a molecular basis for what we knew clinically all along, that males are more likely to withdraw from in utero exposure and suffer from NAS. This work continues as we continue to understand this reward signaling implications. What does this mean for the future of these babies? Has this been a permanent disruption, and how is this occurring? We've had 2 recent papers last year in Frontiers in Pediatrics, last month in Pediatric Research, Also looking at the role of inflammation and cytokines in these molecular pathways. All of it is being conducted in saliva. All right, I'm going to end with perinatal applications, and that's going to be preeclampsia. So preeclampsia is a hyper disorder of pregnancy estimated to affect 2 to 8% of all pregnancies and causes up to 40% of preterm births worldwide. It's a leading cause of maternal and perinatal morbidity and mortality, affecting 60,000 mothers globally and results in 500,000 deaths annually. You make the diagnosis of preeclampsia at the time of disease onset, and we have limited understanding of who will and who will not develop the disease. Our treatment options are limited, and cure is actually delivery of the infant, regardless of age. There are two types of preeclampsia. We classify it as early onset, less than 34 weeks, or late onset, greater than 34 weeks gestation. The earlier you have onset, it's believed to be due to abnormal placentation. Late onset is believed to be due to underlying maternal endothelial and vascular disease. There are serum biomarkers that can have been associated with preeclampsia. They are listed here. They're all involved in abnormal placentation and vascularization. I was actually asked by NIH to see if I could do something in saliva with this. And so I said, all right, I'll try. And I went for two. I went for placental growth factor and placental alkaline phosphatase. So placental growth factor is increasingly important molecule in predicting, diagnosing, and treating preeclampsia. It's proangiogenic, and it works at the fetal-placental circulation and supports trophoblast growth. Its low circulating levels precede the manifestation of clinical disease in preeclamptic pregnancy and growth restriction. PLAP is interesting. It's actually a protein-specific marker found on exosomes. And PLAP-positive exosomes are seen in maternal blood as early as the first trimester, and then they increase throughout gestation. And levels of these have been known to be increased in pregnancies affected by preeclampsia, as well as other morbidities. So we again did a pilot study. It was a small study. There were 14 women with preeclampsia and 15 women gestationally age-matched controls. These women had preeclampsia at time of enrollment. Some were early, some were late, and we just quantified the biomarkers. We actually didn't know if they were in saliva at the time. And I actually partnered with another colleague of mine, Dr. David Wong at UCLA. He ran the assays. Unlike babies, women can spit all day long. It's not a problem getting it. We actually got a whole teaspoon of saliva from them. We did do the supernatant. We stabilized it, shipped it to Dr. Wong out in UCLA. We collected some pertinent demographic data, including gestational age, maternal BMI, and we just used simple ELISAs to measure PLAP and placental growth factor, and we did some just basic statistical tests. And what we found was that first and foremost, we could detect them. They were present in saliva. We published this back in Placenta in 2019. PLAP was really no different between cases and controls, but placental growth factor levels were different. Cases, as predicted, were lower than the controls. And we ended up splitting these out, and we saw that what was really driving the significance was late onset. It was greater than 34 weeks gestation. So two important findings: salivary placental growth factor mirrored plasma levels. We saw lower levels in women with preeclampsia. But importantly, placental exosomes were detected in maternal saliva. I mean, just thinking about that's crazy. But there they are, these placental biomarkers. So we have an important opportunity to integrate these technologies into maternal care too. They're not just for babies. And so even though NIH asked me to do these studies, they didn't end up funding it, but industry did. So we are right now conducting a prospective observational study on salivary biomarkers during pregnancy at Women and Infants Hospital to determine if saliva can predict impending preeclampsia, not at time of disease, based on biomarker profiles. We collect saliva in the clinics on a regular basis across all trimesters. Moms say yes, they spit in a tube and they're out the door, and then we're going to correlate them with pregnancy outcomes. So stay tuned for that as well. All right, so I hope I've convinced you it's applicable. Perinatal salivary analysis has a really broad range of applications, from development, infection, impact of opioids, preeclampsia, diagnostic platforms. But two things are important. You have to do due diligence and scientific approach in order to really demonstrate accuracy and reproducibility of any assay. This is labor intensive and tedious work. And so much of my publications in SPIT have been on technical challenges associated with working with this biofluid. Specifically in newborns, you have to think about contamination with breast milk and blood. And considerations for saliva as a biofluid are also beyond that. Your mouth is filled with microbes. And if you're going to use a platform like RNA-seq, you have to know how to handle those microbes. We have published this. This is another paper out in 2020 on what to do and how to account for microbes so they don't interfere with your analyses. And you have to use stable reference genes and proteins for normalization. And don't forget that you can't use varying volumes because saliva flow and saliva concentration is dependent upon hydration, just like your urine can get concentrated if you feel dehydrated. The other question I get asked a lot is saliva versus blood. I get asked for, well, what's the DRD2 levels in blood, for example? And I say, I actually don't care. No one's ever looked. I'm not looking now. And I'm only asking the question, what's the DRD2 levels in saliva? And is it informative? So if you were to integrate these platforms, ask yourself 2 questions of whether or not you need to do a blood comparison. If existing normative values exist for that biomarker in blood, there's a gold standard, and I'll pick glucose because it's easy. If you're going to tell someone I have an assay and